Good afternoon, everybody. Now we begin to have a first session dedicated to renal diseases. And my name is TRUONG from Houston Methodist Hospital. It is an honor for me to serve as the moderator for this session. This session will accommodate 4 different talks. The first one is related to the structural electron microscopy of the tubular cast. The second one is the utilization of electron microscopy to diagnose monoclonality of light chain. The third talk is electron microscopy of unusual glomerular lesion. And the last one is the utility to diagnose its very distinctive new entity, podocyte infiltration in the invasion glomerulopathy. I'm sure that the talk is going to be very informational, and it provides updated information, but also point out the controversy which can serve as a seed for additional study. I'm sure that this talk will initiate a lot of comment and question, but at the request of Dr. Guillermo Herrera, we may wish you to hold all the questions and comment to the end. So now here I am. I'm excited to give the first talk on this particular topic, and I promise you that the talk is going to be very simple. We just show you a few pictures of light microscopy, and after that we show the electron microscopy, and that's it. So first of all, the definition, the tubular cast is basically a structure formed in the distinct convoluted tubules and connecting duct. It composed of material filling the tubular lumen and molded along its contour. The cast component is composed of basically molecule or structure passing through the glomerular capillary infiltrate. In addition, the molecules secreted by tubular cell or tubular structure can also be the component of the tubular cast. Very broad morphological spectrum. Sometimes it's very frequent and quite ubiquitous. Some others are very rare. Now the morphology, the nature, and the significance of these casts are usually very well known by light microscopy and immunofluorescence. And they're very limited EM finding, and probably because nobody needs to know the ultrastructure feature of the tubular cast for either diagnostic or prognostic purpose. But then we need to know something about its ultrastructural pathologies anyway. So there are so many casts. I just wanted to show you basically the type of cast I will quickly go you, including uromodeling cast, white blood cell cast, variolar cast, red blood cell cast, pigmented cast, hemoglobin cast, myoglobin cast. And then we'll also talk about somewhat unusual findings. That polyclonal light chain, what I call polymorphous cast, polyclonal light chain crystalloid cast, monoclonal light chain cast, also called myelomeric cast, and monoclonal light chain crystal cast, monoclonal light chain amyloid cast, paraneoplastic chromogranulin cast, calcium oxide crystal cast, calcium phosphate crystal cast, vancomycin cast, and amyloglobulin cast. These include a majority of the tubular cast that we encounter. So here are the examples of the uromodeling cast. Basically, it looked like this. It's basically composed of the material secreted by distal portion of the nephron, and usually it is secreted in the urine. However, if we have acute tubular injury of the nephron, then these molecules accumulated in the lumens of the tubule and create casts like that. That is the typical H&E picture. Pale material, strongly PAS positive, and that is the very transparent trichrome stain, and the silver stain, it shows a variable amount of the silver stain positivity. And when you do the immunohistochemical staining for uromodulin, it's strongly positive. And that is the way the thick section would look like, and that is the way electron microscopy look like. It's basically composed of fibrillary materials arranged in different intensity. Some area is a light density, some area is stronger intensity, and this corresponds with the light color here and stronger color here. And high magnification certainly show the same structure. And sometimes there's included a cytoplasmic fragment. And basically, this cast is very frequent. It is indicative of acute tubular cell injury. And most importantly, it serves as the needles on which practically all the other types of cast will establish. The next one is the top cast, is a high-end cast. Every renal pathologist see that in practically every kidney biopsy. And here is the typical HA picture. The strongly PAS positive with some variegated appearance, probably because the different amount of uromodulin accumulated in that, and probably there's additional component in addition to uromodulin also, and that's the typical trichrome stain like that. Importantly, the silver stain is completely negative. When you do the immunofluorescence, typically strongly positive IgA, strongly kappa light chain, lemma light chain. Sometimes we have IgM, lots of times there's no C3, and usually there's no IgG. So the constant component is IgA, kappa, and lambda. So let's see how electron microscopy look in this situation. So that's what electron microscopy would look like. As you can see here, that is the thick section reflecting the morphology we see in light microscopy. And here it's composed of variable structure with variable density, reflecting the life of the duration of cast formation and probably the concentrations of the uromodulin. For example, that is the lighter area, the darker area. In that area, that is even darker and darker. In addition to that, aside from the structure consistent with uromodulin, there seems to be other structure would probably represent cytoplasmic fragment. Now, because they're very strong immunoglobulin, IgG, IgA, and lemma kappa, we have to say that some of the structure probably represent light chain deposition and immunoglobulin deposition, and perhaps all the tiny dots that you see here may represent the deposition of light chain as well as the immunoglobulin. We come back to that pattern a bit later. The next type of cast is the white blood cell cast, as you can see here. It's the typical one. There's accumulations of necrotic or intact neutrophil within the tubule. And when you do the PR stain, obviously there is strong PR stain in the background, suggesting that in every case like that, there's also a background of uromodulin. The neutrophil does not freely accumulate it in the tubulin. It requires uromodulin as the needles. I suppose there is another one here with also pure uromodulin cast for comparison. When you do electron microscopy, typically there are fragmented neutrophil, as you can see here, but interestingly is that there's a background here. This background here is composed of pale, freely material, typically for uromodulin depositions. Another type of cast that we can see is the granular cast. Basically, it represents the situation in which there is extensive degeneration of the neutrophil. Now, we don't see cellular structures anymore. We see granular material according for the name of granular cast. And here is the thick section. Let's see how electron microscopy would look like. And here's what we see. Again, there's all this material in low magnification, high magnification here. It's not difficult to speculate that all this material represents cytoplasmic fragment that belongs to the degenerated neutrophil again. That is a background of fibrillary material representing a background of uromodulin. And the next type of cast we always talk about is the red blood cell cast, as you can see here. That is the typical pictures of that, in which we have probably degenerated red blood cell, and we have intact red blood cell, and this is the features of the PAR stain, that is trichome stain. When we do immunohistochemical study for glycophorin A, which is the marker for the cell membrane of the red blood cell, they are strongly positive, and that is the typical picture that we see is the interleukin blue. In some situation, the content of red blood cell disappear, and now we have only the membrane of the red blood cell to be visible. That is the electron microscopy of the cast, as you can see here. There is a tubule would probably represent distal tubule, and these are degenerate red blood cell. Now we don't see the hemoglobin content anymore, and now only the cell membrane red blood cells are retained, and here are the typical features of that. Again, interestingly, there is a background of uromodulin. Another type of cast that we very could see is the pigmented cast. I see them all the time and always ask myself, what is the nature of that? And when I look in the literature about electron microscopic features of this type of cast, I could not see any. So I tried to identify some of these casts and do electron microscopy on them to see what it looked like. So that is the way the light microscopic feature look like. That the material with a light pigmentation, you can see here, that the PAR stain, that is the trichome stain, indicated there is some red blood cell in there, and that is silver stain. On immunohistochemical stain, these casts stain strongly for glycophorin. They are negative for hemoglobin. They are negative for myoglobin, and that is what it looked like. And that is seen in the case of post-reposition transbiopsy. So we see them all the time, and I ask myself how the cast look like by electron microscopy. So that's what we could find. This is the thick sections of corresponding one. Even with the lutein bleed, I would like to convince you that there's a little bit of pigmentation within this cast here. And that is the electron microscope equivalent of that. And of course, they are composed of red blood cell. And now that high magnification, interestingly, in contrast to the red blood cell cast, sometimes in some red blood cell, although the hemoglobins are gone, there's some infrastructure like that. I wonder whether or not it is these granules here that impart some kind of pigmentation to the what we call pigmented cast that we see here. Now we come to another type of cast, is the hemoglobin cast, and it is the case of renal biopsy in the case of intravascular hemolysis. And then the typical features on H&E, it have a multi-lobular, small granular structure. Some of them are pigmented and some of them are not. The PAR stain delineate the appearance of the cast, but does not tell you the nature of that. Typically, the trichome stain are very strong, but in fact, the trichome stain in our experience is quite variegated. When you do immunohistochemical for hemoglobin, they're strongly positive like that, and we could catch in the thick sections a hemoglobin cast, and on the structurally it looked like that. You can see what I can see: irregular geographic structure composed of dark, very, very dark granules of fibrillary, and that's a higher magnification of that, reflecting what we see in the light microscopy. Again, interestingly, the background of this is the uromodulin material. And this is the case of myoglobin cast. This is a case of myoglobin cast identified in the kidney biopsy in a patient suffering from acute kidney injury during a cross injury. Of course, we all know that there are many etiology for the formation of the myoglobin cast, but here we focus only on morphology. On H&E that look like that, again, that have irregular geographic structure, some type composed of microspherules in aggregation in irregular configuration. And again, typically it is formed in the distal portions of the tubules. And that is another picture of the H&E, in which we have now pigmentation in contrast to the poor pigmentation here, the PAS stains are here, and the trichrome stain here. As you can see, the light microscopy is practically identical to the hemoglobin cast, and that is the silver stain completely negative. However, when you do immunohistochemical staining for myoglobin, they're strongly positive. And that is the thick section. That's what it looked like. And this is electron microscopy of the material. Again, you can see that there is the geographic aggregations of the material with either short fibrillaries or granular, and it have a different intensities and density. And when you go with high magnification, some of the structure show that kind of laminar configuration, as you can see here. And again, the typical background of the uromodulins. Now, this is an interesting case that I'm not sure that has been emphasized in literature. In several situations, including the poultry perfusion biopsy, we see some structure like this here. It looked like this one. It looked like this one. And I thought that, okay, it may be myoglobin, but when you do immunohistochemical staining, the myoglobin, hemoglobin, and all the other, it's completely negative. But when you look at the immunofluorescence, they are strongly positive IgG, IgA, kappa, and lambda. And we are lucky to get them in the thick section. So we submitted for electron microscopy. And that is another similar situation, which I don't think that has been emphasized in the literature. And this is the case of the intraoperative wet biopsy from a donor. And what you see, some structure like that. Initially, I thought that it's the necrotic cell, but does not look like one. We do the PAS stain, it look like one. And we do the immunofluorescence, it's also strongly for all kind of immunoglobulin, including lambda and kappa. So this case is quite similar to the previous one. So we go ahead and do electron microscopy on both of them, and that's what we see. We see material like this here. It got, again, this structure here, distal tubule, and there is a very dense granulated material. And this was the time I guess the doctor Herrera would tell the okay one, that is the light chain molecule, but it's the polyclonal. So you can see here and you can see here. It's composed of very elegant dense, tiny granular material that reminiscent of people decide that granular deposit along the tubular basement membrane, glomerular basement membrane, typically in light chain deposition disease. So I submit to you that all these material represent polyclonal light chain depositions. Now there's another case that I think that we see that not infrequently, but not very well described in literature. Here is the H&E biopsy. When we look at that one, you look at PAS, look at trichrome stain and silver stain. It is a very reminiscent of myoglobin cast on hemoglobin, except that all the stainings are completely negative. And I think that it looked like a crystalloid materials also. But let's see how electron microscopy look like. And in fact, we are lucky to capture that cast in the fold section. And when you do a fold section, the strong kappa light chain, strong lambda light chain, and also the other type of immunoglobulin. And that is the thick sections here. I venture to say that these casts here look quite similar to myoglobin cast or hemoglobin cast. But when you do electron microscopy, it looks just like that. That is the typical features of light chain deposition. There are the isolate particle material here, and sometimes it aggregates into very dense geographic configuration that we see here, and there's a high magnification of the structure. So in my conclusion is that there are situations in which the light chain deposition can appear as crystalline material, but it's still polyclonal, not monoclonal. The point that I think Dr. Guerrero will mention later. Now we come to the one that all of us are quite familiar with, and that is the monoclonal light chain cast, also called myeloma cast nephropathy. So that is the typical H&A picture in a PAS, typically negative in trichome stain, quite variegated, and trichome stain in orange color, blue color, every renal pathologist is familiar with that silver stain. And when you see, we do fold section, it is always monoclonal, as opposed to the two casts I will show you before. And that electron microscopy feature of that, and in my opinion, that look practically identical to the polyclonal light chain cast. And here is another appearance. In this particular case here, there is the geographic configuration, and there's also the cell associated with that, and the fracture area here. And I'm sure that Dr. Graham will tell you more about that. And it's suffice to say that some electron microscopic feature of this cast can differentiate that monoclonality from polyclonality, which are very important considerations. Now, there is some variants of monoclonal cast, and there is 3 variants of them, and let me show each of them. One of them is the crystalloid cast formation. You can see them right here, and that is typical morphology here, typical morphology. Trichome stain look like that, silver look like that, and very strong monoclonality. It is very important to recognize that if you do the immunofluorescence with this type of cast, it is almost always negative. However, if we perform the immunofluorescence staining for the peripheral tissue, then the monoclonality is readily revealed as this one here. As exemplified by this case, the immunofluorescence for section completely negative, but when you do immunofluorescence and peripheral tissue, it is clearly monoclonality. And that is on the structural equivalent of this type of cast. Another somewhat unusual forms of monoclonal cast is the monoclonal cast forming an amyloid formation. Then that is the typical feature. Again, it looks quite similar to the other type of cast, the myoglobin or hemoglobin. But when you do converse, that is strongly like that, and it is strongly enhanced by Texas rat. And we do lambda kappa light chain in consecutive section, that is the monoclonality, and that is thick section, and then electron microscopy. I venture to say that, well, these structures here have the typical fibril structure, typical amyloid. And interestingly, aside from that, is all the background of what looked like uromodulin. So that's also featured of amyloid monoclonal cast. Now we come to another type of cast. There are situations. Now, how much time I still have? Almost done. Well, I mean, I'm almost done also. There are situations in which the tumor will make a lot of the paraprotein, and the paraprotein in circulation somehow accumulated in the tubule to form a cast, and that is called a paraneoplastic cast. The chemical structure of the cast is variable. Most of the time, it represents chromogranulin A because this type of casts are more frequently seen in metastatic neuroendocrine tumor, and that is the example for you. So that is the cast here. That is trichome stain. That is silver stain. That is the image stain for chromogranulin. And that is the typical electron microscopic features of that. And now we have about 3 more type of casts to show you. Then that is the calcium oxide crystal cast. By actually look like that, and polarized material look like that, actually look like that. There are 2 forms. One is irregular form and the other fan shape. Everybody know how this one. That's why nobody cares about electron microscopy. So we look carefully in our file, and that is the one morphology up the kind of round cast, and that is the morphology of the fan shape cast. Here's another one. That is the calcium oxide cast material. It looks like this here, and they have wider structure here, bronchosustain, thick section. But when you look at them, there's a very interesting structure. The most interesting structure is the fact that it's a tiny needle-like structure like that, which would form very conspicuous speedo structure. We have seen several cases, and in every case, the calcium oxide crystal would look like this. Now, the next type of cast we see quite frequently, and I think it's a very characteristic, almost pathognomonic for disease, is a vancomycin cast. We see this very frequent. We see about maybe 10 cases per year, 15 cases per year easily. Actually, it looked like that. When you see uromodeling, the strong staining, but when you do image staining for vancomycin, it's also positive. When you do electron microscopy, there is a very characteristic structure, and some of them the beautiful laminated material like this. And finally, that is the case of aminoglycoside cast. Now, usually, this is a cast, tubular cast material with a structure like that. And when you do electron microscopy, all the structure turn out to be myelin body. Now, most of the time, this is within the cell cytoplasm, but in some situation, it can get a tubular lumen. It combined with uromodeling to form tubular cast like that. So I think that. So that is the last type of cast I want to share with you. And just some conclusion, the EM study of renal tubular cast really add to novel information. And under my eyes, it looked quite beautiful images that just it on my own. The EM finding should corroborate traditional morphology, and the EM can facilitate a definitive diagnosis. And in fact, sometimes EM study may add to diagnosis and pathogenesis. So that's all I want to tell you about today about the tubular cast. I'm sure that there's a question, comment, but we will wait to the end of the session.